Every microscopic entity on Earth faces the exact same engineering problem: how to protect fragile genetic code from a highly hostile environment. Eukaryotic cells solve this through strict internal compartmentalization. They build membrane-bound organelles to isolate volatile chemical reactions and physically wall off their DNA inside a highly regulated nucleus. Prokaryotes strip away that internal complexity. Bacteria rely on absolute structural simplicity and a rigid outer envelope to survive, an architecture that requires far less energy and allows for rapid replication. Then there are viruses. They carry no organelles and build no physical defenses of their own. As obligate parasites, they bypass the need for independent cellular infrastructure entirely. These three distinct structural strategies dictate exactly how we treat infectious diseases. If we want to neutralize a pathogen, we have to know exactly how it is built. A bacterial cell contains 0 membrane-bound organelles. Its genetic material and ribosomes float freely in the open cytoplasm. Without internal barriers to isolate damage, a bacterium relies entirely on its outer perimeter. Its survival depends on the structural integrity of its envelope. This shows the defensive barrier of a gram-positive bacterium. Notice the thick, rigid peptidoglycan layer, a dense polymer mesh held by cross-linking bonds. Penicillin targets these bonds, wedging into the crosslinks during cell growth, fracturing the peptidoglycan and causing the wall structure to fail. Now look at a gram-negative cell wall. The peptidoglycan layer is much thinner, actively hidden beneath an outer membrane. Extending outward are lipopolysaccharides. This layer acts as an effective barrier against many drugs and triggers a severe inflammatory immune response in the human bloodstream. Because a bacterium cannot retreat behind internal walls, destroying this rigid envelope strips away its only physical defense. The cell lyses and the organism dies. Bacteria reproduce through binary fission. They copy their single circular chromosome and divide, yielding identical clones. If one cell is vulnerable to an antibiotic, the entire colony is vulnerable. To survive a chemical attack, that uniform population must rapidly acquire genetic variability. They achieve this without sexual reproduction by sharing DNA horizontally. The first method is transformation. When a neighboring bacterium ruptures, it spills its internal contents. Nearby cells sweep up that free-floating genetic material and integrate it directly into their own genome. The second method is conjugation. Two living bacteria extend a fragile bridge, called a sex pilus, to connect to one another. Once linked, the donor cell pushes a copy of its DNA directly across the bridge into the recipient. The genetic material transferred is usually an extra chromosomal ring of DNA known as a plasmid, such as the F factor, which grants the recipient the ability to form its own bridges. These plasmids frequently carry specific virulence factors, or genetic instructions for antibiotic resistance, like efflux pumps that physically eject drugs from the cell. A third route is transduction. Here, a bacteriophage virus accidentally traps a segment of bacterial DNA, then injects that genetic material directly into its next host. By continuously swapping plasmids and loose DNA, a static colony of clones transforms into a dynamic, interconnected network capable of deploying a shared chemical defense. This graph models that exact scenario. The y-axis represents antibiotic stress, while the x-axis tracks the growth efficiency of the resistant cells. Look at the bottom edge, where the antibiotic stress is 0. In a drug-free environment, the sensitive strain dominates the population almost entirely. The resistant strain only holds its ground at the extreme far right, at a 1.0 maximum. Now raise the antibiotic stress to a highly lethal level. Notice this enclosed area that emerges on the graph, labeled competition-induced extinction. If the resistant strain's replication speed drops slightly, the sensitive strain briefly spikes above the resistant cells, aggressively outcompeting them for resources. But the sensitive cells require the resistant cells to deactivate the antibiotic in the environment. By starving their only chemical shield, the sensitive cells doom the entire colony. Both populations crash to absolute 0. The only way the colony survives high antibiotic stress is within this narrow zone of co-colonization. The math reveals a strict requirement. The resistant strain must maintain a strict growth efficiency strictly lower than 1.0, replicating slower than the sensitive cells it shelters. Collective resistance requires the resistant strain to sacrifice its own maximum growth rate. By slowing down, it diverts energy to deactivating the antibiotic, maintaining a shared ecological shield that keeps the vulnerable cells alive. Bacteria wage wars of chemical attrition at the cell wall. But another class of pathogens renders physical architecture entirely irrelevant. Viruses are obligate intracellular parasites. They possess no ribosomes, carrying zero machinery for independent protein synthesis. A standard virus injects its genetic material, like positive sense RNA, straight into the host's cytoplasm. The host's own ribosomes read this foreign code as if it were natural messenger RNA, immediately assembling viral proteins. Retroviruses, a specific family of enveloped, single-stranded RNA viruses, take this subversion a step further. This diagram shows a specialized viral enzyme called reverse transcriptase. It reads the single-stranded viral RNA and forces the construction of a matching, double-stranded DNA helix. A second enzyme, integrase, splices that newly forged viral DNA directly into the host cell's own chromosomes. The host cell's machinery now permanently holds the instructions for its own parasite. It blindly transcribes the code and manufactures mature virions, seen here budding outward from the cell membrane to infect new targets. Once inside, viral reproduction follows two potential paths. The lytic cycle prioritizes overwhelming force. The virus forces the host to produce massive numbers of new virions until the cell ruptures. The alternative is the lysogenic cycle. The viral DNA remains dormant within the host genome. Every time the host cell naturally divides, it silently and safely replicates the viral code along with its own. Viruses do not try to breach the cell wall with blunt force. They weaponize the host's internal blueprints against itself, ensuring the infection is perfectly masked by the cell's normal operations. The eukaryotic cell survives by relying on heavy internal compartmentalization, physically isolating its critical biological functions. The prokaryote discards internal complexity to rely entirely on a rigid external envelope, surviving chemical attacks by sharing defensive plasmids across a local population. The virus bypasses structure completely, subverting the core genetic processing of a host to force its own replication. Modern medicine is the practice of mapping these precise structural boundaries. Whether it is deploying penicillin to fracture a bacterial cell wall, or administering antiretrovirals to jam a viral enzyme, treatment relies on engineering the exact chemical wedge needed to break them.